In this lecture, I am going to talk about immunity to helminths. Helminths are multicellular, not intracellular pathogens, and accessible to the immune system. But however, levels of immunity generated to helminths is often very poor. And I will highlight some of the worms that affect humans and animals. For example, Ascaris round worm that infects the small intestine, Schistosoma species. For example, Schistosoma mansoni, Schistosoma japonicum, and Schistosoma hematopian. So helminths, domestic in animals and invade humans, include tinea, tapeworm of cattle and pigs, Trachinella. So in helminthic infection, interleukin-3 from T helper 1 stimulate growth of mast cell, and as can be seen here, this is stimulated mast cell. So mast cells, they produce granules. These are platelet activating factor, eosinophil chemoattractant factor, neutrophil chemoattractant factor. So localized increases in mast cells at the site of infection and their subsequent degranulation, as can be seen here, increase the infiltration of macrophages and eosinophil in helminthic infection. And also T helper 2, IL-5 or interleukin 5 from T helper 2 induces bone marrow precursor to differentiate into eosinophils. These are eosinophils. So eosinophils, chemoattractant factors from mast cell recruit more macrophage, more eosinophils, and specific IgE antibodies, as can be seen here. These specific IgE antibodies raised against antigens on the surface of the worm. It binds to the specific antigen on the surface of the worm, and it binds to Fc epsilon R1 receptors on the surface of the eosinophils, and these are actually high affinity antibody, and these are receptors. It is high affinity receptors on the surface of eosinophils. So this interaction between the eosinophil and the antibody and the worm, it leads to release of granules from eosinophils. And as can be seen here, these are granules from the eosinophil following the interaction between the antibody, IgE antibody, with eosinophil and the specific antigen on the surface of the worm. So eosinophils, they following this, they produce eosinophil cationic protein, as can be seen here, and eosinophil derived neurotoxins, which is toxic to the worms, and major basic protein, which is also toxic to the worm, and proteases. And also basophils take part in immunity to helminths. And as can be seen here, interleukin 4 from activated basophil induce M2 macrophages arginase 1, and this is actually the conventional macrophage. They produce tumor necrosis factor alpha and interleukin number 1. But however, M2 macrophage, they secrete arginase 1 and interleukin number 2, interleukin number 10, sorry. So this interleukin number 10 is very important to ensure T helper 2 immune responses and to downregulate T helper 1 immune responses. So as can be seen here, these are proteases by the basophils. So these proteases recruit monocytes, neutrophils, and more eosinophils to the area of infection. And the classical pathway of the complement also take part in immunity to Helminths through its activation by antibodies, as can be seen here. These are shed antigens from the surface of the worm or of the Helminths, and these antigens will be taken by the antigen presenting cell, as can be seen here. This is a macrophage and B cell, maybe dendritic cell. Then the processed antigen will be presented to T helper 1. Activated T helper 1 will produce interleukin number 2 that activate T helper 2 and B cell. So B cell and T helper 2 will cooperate together and produce antibodies of IgG and IgM. So these antibodies will interact with complement component number 1 C1q, and this will lead to activation of the classical pathway of the complement, and this result in formation of the membrane attack and aflatoxins, which lead to lysis of worms. Thank you very much, Professor Ahmed Boulad.